Okay, everybody, welcome to the Biocom 2021 virtual edition, September edition of the virtual edition of Biocom. I'm Adam Cooper, your president of the BCA, and I would like to welcome everybody here. Today's presentation is from Dr. Maja Divjak, and I said that correctly, right? And so, Dr. Maja Divjak is the inaugural biomedical animator at the Peter MacCallum Cancer Centre, where she's creating animations about cellular and molecular mechanisms leading to cancer. And that is in Melbourne. And my Aussies know that I just pronounced that correctly. So without further ado, I would like to present Dr. Maja, and we welcome her. Thank you.
Thank you so much for that lovely introduction, Adam, and thank you for the wonderful invitation to speak today. It's an honour. I'll just start by sharing my screen. Okay, so my talk today is about biomedical animation, using 3D animation to tell the stories of biology. So I'll just start by giving you a brief overview of where we are going to go today, so you have an idea of what it is we're going to cover. I'll begin with defining what biomedical animation is and why it's important. I'll then cover what a biomedical animator does, and then we'll go into some detail looking at a project outline and the steps that I follow when I'm creating a project. I'll then discuss some of the artistic devices that I use to facilitate understanding and to direct the viewer's attention. And I'll then give you a brief overview of the software that I work with and some of the main tools within that software. And then we'll cover some of the challenges and joys of being a biomedical animator. So let's dive right in. Okay, so what is biomedical animation? Biomedical animation uses 3D animation techniques from cinema, so as you would have seen many times throughout Hollywood films, to explain biology. And in doing so, it accelerates understanding because it unfolds before your very eyes. It engages people with science because they can understand it. And it illustrates the breathtaking beauty of biology, as these three images rightly show. And that's one of the things that gets me out of bed in the morning is that, you know, what I do is bring the beauty of biology to the world, and it's a wonderful world to be part of. So what does it do? What does biomedical animation do? Primarily, it makes the unseen world of molecules and cells visible. At the moment, only the electron microscope is powerful enough to see molecules. And as most people don't even have access to a light microscope, let alone an electron microscope, which is a very large piece of equipment that requires expert knowledge to use, how is anyone going to be able to see this world and know that it exists? So that's what biomedical animation does. It brings that world to life and makes it visible. But then you may ask the question, how do I know what these molecules look like so that I can then visualize them and animate them? So what I do is I rely heavily on data from the Protein Data Bank, which is a repository that at last count contains about 182,000 structures, 54,000 of which are human. And these include protein and nucleic acid structures. And so I'll just give you a little bit of a dive into this world. Much of the data that I work with is obtained by X-ray crystallography. Now, I'll give you a brief overview of what that is. When you're working with proteins, you can generate proteins and crystallize them, and then you can work out their structure by taking them to a synchrotron and then bombarding them with high-speed X-rays. And the way that the X-rays actually hit the molecule and then the trajectory that they take when they bounce off gives you a series of X, Y, Z coordinates in space that defines the shape of that molecule. So you've got the X-axis, which is the horizontal axis, the Y-axis, which is the vertical axis, and then you've got the Z-plane, which is the third dimension. And so these coordinates are deposited in the protein data bank, and then I'm able to take that information, bring it into my animation software, and then animate those molecules. So yes, biomedical animation makes the unseen world of molecules and cells visible. It also allows understanding of new discoveries that might otherwise be really difficult to explain using text or even 2D imagery, so squares and circles on paper. And it can offer insight to scientists. So I work largely with life scientists, and they tend to think about their hero molecules in a kind of abstract sort of way. They tend to think about them in terms of what they do and how they function and how they kind of interact with other molecules. They don't tend to think about their shape or how they fill space and how they kind of move around. So oftentimes seeing their hero molecules in action in animated form can really inspire ideas about what they're doing and how they work. And biomedical animation is a visual presentation of biology that is not only useful because it's educational, it can really be a work of art as well. And again, that's another thing that gets me out of bed in the morning, is that, you know, the result of biomedical animation is really beautiful. And amazingly, it's also the gift that keeps on giving. So the animation itself is not the end game. They can be realised and reimagined in many other different forms. So you can create still images from the animation, and you can bring these to life via your smartphone and an app called Artivive. And if you point your phone at these images and look at them through the Artivive app, the image will come to life because the app recognises that image and then plays an underlying associated animation. You can make interactive iPad resources for patients explaining, for example, cancer biology and cancer treatment, centred around an animated piece of work. You can create holograms from them, which is something I'm working on at the moment with a local contemporary artist. You can project them onto, you know, beautiful wall spaces. And oftentimes, when scientists submit their work to research journals, they are asked to submit cover art illustrating their work so that explains their science. And so often you can take stills from animation and submit it to journals for cover art. So why is it important? As you probably know, 60% of our brain is devoted to processing visual information or performing vision-related tasks. And we're visual creatures, and we have been since the days that we first scanned our environment in search of food. But, you know, these days, with the absolute rise and domination of the internet and the rise and domination of social media, we're becoming ever more visually focused. And so it is therefore one of the best ways to engage audiences. This is a really important one. It connects the research lab to the public, and it does this by giving insight into the research being conducted in labs around the world. As you're probably aware, you know, most people that are not in the science world think that scientists are very strange people who wear white lab coats and do strange things behind closed doors with their tax dollar. And we need to really address this mistrust by opening the lines for communication to better inform what it is that scientists actually do. And biomedical animation actually does this. And so through a process of education and understanding comes a process of inclusion. It removes fear of science because it makes science accessible through visual rather than abstract concepts. And it really gives an appreciation of the beauty and the drama going on in your own body and connects you with your biology. You have a whole universe of cells inside your body, and then you have a whole universe of molecules inside those cells that allow those cells to function and interact and to exist and to make sure that the body functions as a whole. And people are completely unaware of this world. Most people are aware that they have tissues constructed of cells, but they are completely unaware of this molecular world. And all of this just goes on without people or anyone having to think about it. And then sometimes things can go awry, leading to situations like cancer. So biomedical animation really focuses a lens on that invisible world. So why is it really important? So you could say this: DNA damage consists of single-strand breaks, which are discontinuities in one strand of the DNA helix, double-strand breaks where both strands of the DNA duplex are severed, DNA kinks, which occur when stacked base pairs become unstacked, causing DNA to make a sharp turn, and pyrimidine dimers, which cause two separate DNA helices to stick together. And this is the sort of response you're going to get. People are completely confused, they disengage, and moreover, why would they even care? Or you could show this. Here we are.
DNA encodes the information needed to make proteins, which are the building blocks of our body. To keep 2 meters of DNA organised within the nucleus, it is wound around special structures called histones, much like thread on a spool. Damage to DNA can include single-strand breaks, double-strand breaks, changes to the DNA code, DNA kinks, and DNA sticking together. It has been estimated that an individual cell can suffer up to 1 million DNA changes per day.
You can see what a completely different situation that is when you can actually see all those things happening before your eyes. How much easier it is to understand. Okay, so why are visuals important right now in our pandemic afflicted world? Medical illustrators at the Centers for Disease Control, which is the National Public Health Agency of the US, as I'm sure you're all aware, they realised that they needed to create a visual identity for COVID-19 because people didn't know what it was, they had no idea what it looked like, and there were people out there who thought it was a hoax and a complete conspiracy. So, and they knew from previous experience with other pandemics like swine flu and bird flu, that without knowing what COVID-19 looks like, it would be really difficult to communicate the risks to the general public. So they knew that the public needed to see and understand the unknown enemy in order to believe that it was real. And they wanted to create an image that was scientifically accurate, but that could be readily recognised and digestible for a general audience. So we'll go through some of the steps that they followed while creating the spike protein, which is the surface protein on COVID-19 that allows it to bind to the host cells. So in the top left corner here, we can see the PDB file. So this is the information that's deposited in the protein data bank, which I've already mentioned. And so these are the X, Y, Z coordinates that define the structure of that spike protein. And this is what the internal structure of the protein looks like. You can see there are lots of coiling helices in the sort of bottom half to middle of the structure. And then you've got lots of kind of sheets stacked in anti-parallel directions around the top. At the bottom left there, you can see what the spike protein looks like on the surface. And then in the middle, you can see the structure from the PDB file that has been imported into their animation software, which is 3D Studio Max. And then on the right, you can see the final result there. There is the rendered spike protein shown in bright red. And the whole image is one we can see here, and I'm sure you've all seen this many, many times. It's become quite iconic. So although they wanted it to be completely scientifically accurate, where they really took artistic license was with colour. And they really wanted to convey a serious mood to indicate the danger of this virus, of this pathogen. So they used bright red for the spike proteins to give this kind of alarming sense. And then they used a kind of foreboding stony grey for the lipids of the membrane structure. And then they made the envelope protein and the membrane proteins in a kind of yellow and orange Again, bright, but not so bright that they outcompeted the bright red of the spike proteins. And so this style guide, this design guide, they decided to use for all their COVID-19 material to give it consistency, and so it would be instantly recognisable. And they found that by giving the enemy a face, people came to believe that the virus was real and it was dangerous and needed to be taken seriously. Now, the designers, Alyssa Eckert and Dan Higgins, they never expected this image to appear around the world. They thought it might be taken up by a couple of cable TV stations at most. But such is the power of a fantastically designed image. It was so sort of recognisable and, internationally recognisable. And it led the movement to educate and inform the public. And it really has become this iconic thing. So what does a biomedical animator do? Let's dive into this in a bit more detail. So a biomedical animator works at the interface between art and science, using 3D animation to educate about science and illustrate biological processes. And so how do they do that? They take lots of complex scientific data from scientific journals, and then they distill this, they convert it into a story in visual format. And so, of course, it has to be scientifically accurate, but it must have sufficient detail, but not so much that the overall message is lost. So you can really end up down the rabbit hole, you know, completely getting involved in animating pathways and, you know, this molecule interacting with that molecule. And, you know, this can have great interest to scientists in the field, but to a lay audience, that sort of level of detail is probably not of interest and is overall completely confusing, and the message can be lost. So you have to tread that fine line between scientific accuracy and clarity. And of course, the image must be striking to have universal appeal for everyone from the highly trained scientists to the person in the street, to the general public. So let's dive into what it takes to create a biomedical animation and the steps that I follow when I'm creating one. You have to start by defining your animation project. And usually what that involves is selecting the most interesting or cutting edge project that you can find at the time. And so at Peter Mac, I'm really lucky in that we're not only a world-leading specialist cancer hospital, we also have an integrated world-leading cancer research program. So I've got a number of projects that I can select from. And yeah, I'm really lucky in that I can talk to the scientists that are involved with those projects, that run those projects. So I can actually talk to them, get the facts straight, make sure the story is being told correctly, and that I have the molecules correct and so on. So I'm really lucky because I have, you know, a number of projects available at any one time, and the scientists are there to be spoken to, which is fantastic. You then need to do a lot of reading. So you need to be able to get your head around the subject in order to be able to animate it. And so this involves going to the scientific literature and reading lots and lots of journals. And what I find is that writing a literature review at this point really helps me to make sure that I've understood the story and that I've got the process straight. And then often what I will do is I will distill that literature review into a visual format. So I will make a storyboard from it, which is, as you can see in the bottom right corner there, a list of sequential shots outlining what's going to be happening in the animation step by step. And at this stage, you can go back to the scientists and show them this, and they can give you great feedback about, you know, whether you've got the story straight and whether the sequence is correct and so on. So it's an iterative process. There's a lot of back and forth between you and the scientists. At this stage also, it's very handy to write a script. I find that that really helps with the flow of the animation because it helps you time the length of the shots according to what you need to say to explain the shot. So that makes the shots flow really nicely so that you don't have some that are too long or others that are short and compressed. So I find the script really helpful at this stage. Many people can actually animate without writing a script at that point, and they leave it to the end. They seem to have an uncanny ability to be able to time the length of their shots. So my mentor, Dr. Drew Berry, is amazing in that regard. He writes a script at the end. You then need to assemble your models from the protein data bank, which I have discussed in a bit of detail already. And you can do this using a program called Chimera or Chimera X, or you can do it within your animation software, which I do. So an example is shown on the left there. That's a model of the EGF receptor and associated underlying membrane molecules. And that's been built in Chimera. And then over on the right, that's the same situation. So the bottom half there in mauve, pink and blue. And that's in the animation software Maya. So you need to assemble your models from the PDB. And oftentimes you'll find a little bit here, a little bit there, and bits here and the missing and so on. You will never find a complete assembly in the PDB. You have to kind of construct it yourself from the pieces, and your reading will inform you as to how to do that. Once you have your models constructed and you've got them in your animation software, you need to rig them. And often this involves constructing a skeleton out of joints and bones so that the models can articulate in much the same way that our skeleton does. There are other methods as well. I often use a system called cloth, which is used for animating clothing on characters. But I find it gives really nice organic fluid deformable motion. So I use that often as well. And then you need to animate them. So you need to apply force to make them move and interact with other molecules. You can then add pretty textures and colors. You light your scene, you create cameras and camera movement. So it's a lot like being a digital director. And the bottom right here, actually, as you're going along and you're checking the progress of your animation, often what you can do is these really rough and ready play blasts to make sure that the animation is proceeding how you want it to and it's behaving correctly. So I'll just quickly show you what that looks like. So this is a portion of the scene shown above there. I'll just get the play button. There we go. So we can see we've got this molecule in orange coming into the KRAS there. It's bound and then it's switched it off and then it's moving away. And then you can see this cage-like structure I've got, which is controlling the lipid bilayer as well. So that gives you some idea of this sort of inner workings of the animation process. And then you come to the process of rendering. And this is the process of building each frame pixel by pixel. And it's quite labor intensive and can be the most lengthy part of your process. Each second of animation, in my case, consists of 25 frames, and you have to build those frames pixel by pixel. And yeah, it can be quite time consuming, depending on the complexity of your scene. You then have to take those frames and put them all together in a program such as After Effects in a process known as compositing. You can also use Final Cut Pro or Premiere or any of those kind of editing programs, which I'm sure some of you are probably familiar with. And at this point, you can edit, add special effects, add labels and captions to actually indicate particular molecules that you're interested in pointing out. And then we go on to the final stages of adding sound effects. And I've worked with a sound designer, Adam Hunt, for quite some time now, about 7 years. And he creates all the kind of pops and whirs and whistles and squeaks and those kind of, clicking sounds as molecules interact. And I find that this is a really important component of the animation. It really brings it to life, and it also directs the viewer's attention and facilitates understanding. And finally, you add narration. You add your voiceover narration, and I've worked with Dr. Claire Fideli for a little while now, and she's fantastic. She gives a level of gravitas to the whole production. So let's discuss some of my artistic approaches when creating an animation. So this one is really important. The story dictates your design. So what you're trying to say really needs to determine the look and the feel of each shot and not the reverse. So you don't design the shot based on how pretty it is if the story is not clear. So as Pixar are fond of saying, story is king. So what you're trying to say really kind of informs the look of each shot. Camera movement and lighting is really important because it can direct the viewer's attention, and you can actually use camera movement to take the viewer on a journey from point A to point B. Choice of colours. This one's quite contentious. It's a little bit controversial. A lot of molecules are not inherently coloured. Of course, there are many, many exceptions to this. For example, indigo, which is bright blue, or ferrocene, which consists of iron atoms and is therefore a rusty brown colour. Or of course, our classic favourite green fluorescent protein, which is a very commonly used genetic reporter. But yes, many molecules are not inherently coloured. So there are purists out there who insist that really you should be animating entirely in grayscale. And there is an Italian biomedical animator called Dr Monica Zoppa, and she works entirely in grayscale. And her work is really quite amazing in that she's somehow able to direct your attention when she's working entirely in shades of grey. And her work is really quite beautiful. You should take a look at it. But I really like to use colour. It's a signature of my work. I like to use it to indicate disease states. So I can tend to use warm pinks and reds and so on to indicate, for example, inflammation. When I'm trying to animate cell death, I will often use murky yellows and browns. And these states often register subconsciously with the viewer. They just inherently know what it is that you're trying to say without particularly being conscious of it. And I also use colour to define different functional parts of molecules. So over on the right there, lower right, that's the BRCA1 assembly. And I've used different shades of green just to delineate the different functional motifs of that protein. And it's completely arbitrary. I just chose green because I thought it looked beautiful in that situation. Yeah, so often I choose colours because I find them beautiful. But in the case of animating cancer biology, there's a bit of a conflict for me because is it really appropriate to make something that people find uniquely terrifying look beautiful? I've often struggled with that. But what I'm hoping is that the colour, although it's beautiful, actually makes no comment on the relationship of cancer to human beings, and that it highlights the complexities and the patterns of the biological molecules. I use glows quite a lot. They're very handy for highlighting cellular and molecular interactions. So as two molecules come together, I'll often have a flash of light, and that will direct the viewer's attention. And yes, sound effects, which we've touched on briefly. Very important for directing the viewer's attention as well and promoting understanding. And not a secondary consideration. As I've explained, I find the sound effects are an integral part of the animation and they really bring it to life and they're very important. So let's take a look at some of the technical aspects of the software that I'm working with. I use Chimera and Chimera X sometimes, not so much anymore, but you can use this to assemble protein fragments from the protein databank into complete structures. As I explained, you will never find complete molecular assemblies in the protein databank. You will have to put them together yourself. And you can use Chimera or Chimera X to do that. Or you can use the animation software, which is what I do at the moment. I use Autodesk Maya, which is industry standard animation software. It's throughout Hollywood and it's fully customizable with scripting. So if you're conversant with Python language, then that will stand you in very good stead. However, if you're not, like me, I don't use Python a whole hell of a lot. I do use it occasionally, but there's a graphical user interface. And so if you're not conversant with scripting language, that's fine. I use a wonderful plugin called Molecular Maya by Clarify. And this is fantastic because it allows you to import structures from the PDB into the animation software in atomic, surface, or ribbon formats. It also allows you to build protein and nucleic acid structures, which is what I use it for a lot as well. It allows you to rig them, not using joints and bones, but using a particle system. And it also allows you to build and rig double-stranded DNA, which is very handy. And I use Adobe After Effects for compositing, editing, special effects, glows, labels, and then animated arrows and pointers and captions and so on for the final touches. So I'll go into some detail now into the Maya Dynamics engine. And so this is a set of tools within Maya that uses the rules of physics to simulate natural forces. And so it allows you to create realistic motion without traditional keyframing. You can specify the actions that you want the object to take, and then the software can figure it out for you. And so I use Maya Dynamics a lot. I've used it in my latest animation about CAR T cells, chimeric antigen receptor T cells. I've used it in my previous animation, What Goes Wrong in Cancer, and in a much older animation about whooping cough. So I've used particle systems a lot to create atomic structures. I also use it for animating lipids in cell membranes, and it's fantastic for generating release of chemical mediators into the surrounding medium. As mentioned, I use cloth systems a lot as well. Yes, these are used to animate clothing on characters, but really handy for biomedical animation because it gives molecules this really lovely organic fluid motion and it makes them soft and deformable. People generally have this idea that molecules are kind of like concrete and they kind of smash together, but that's not the case at all. They're actually soft and deformable. So cloth is fantastic for animating in this way. And I use hair systems, again, for animating hair on the heads of characters. And this is great for giving long rope-like structures whipping motion. And the good thing about these systems is that because they all come under the one umbrella, they interact and collide. So they all communicate with each other, which is fantastic. So let's have a look at where I've used some of these processes. I'll just explain briefly what's going on here and then I'll let you have a look at it. So what I've done is I've created two planes representing each side of the lipid bilayer. And then I've animated the planes with a particle system that's driven by a turbulence field to give jittery undulating motion. And I then applied some animated lipid models to those animated planes to give the very dynamic membrane. And then the molecules lying under the membrane, so those in pink, mauve and blue, they've been animated with cloth and that gives that nice organic motion and the softness. So let's take a look at that.
A regulating molecule normally keeps RAS switched off. To turn it on, RAS interacts with a large molecule in the chain, which opens it, allowing the regulating molecule to leave and another molecule to enter. RAS is now active.
So yeah, used a lot of particle systems there and cloth. And you can see that the molecules are actually represented in surface format there. So in this next animation, here I've used atomic representation. And this is actually an animation showing the surface of a T cell in blue, and it's interacting with an abnormal cell in yellow-brown. So this is from my latest CAR T animation. This is hot off the press. No one's seen it before, but it has no sound. So in this case, I've used particles to generate the atomic representation instead of the surface look that you saw in the previous piece. And I've created the cell membrane in the same way as previously, but this time I've used different lipid models to give a more atomic look. So let's take a look at this. Now we're below the cell membrane. And you can see there what a difference sound makes. With no sound, it really doesn't have the same effect, does it? It's quite interesting. It doesn't have as much life. Okay, and so this final piece that I'm going to show you is an animation about whooping cough. And here we can see the ciliated epithelium in the trachea brushing mucus and trapped foreign material out of the airways. And so I've animated hair systems in this case to illustrate a very slowed down movement of cilia, which was based on a paper that analysed the physics of the forward and the backward stroke of the cilia. So let's take a quick look at that. And you can see there just what the effect the sound is having. We have this saying in animation that gross is good. So all those horrid sucking noises of the mucus, it really sticks in people's minds. So it's important. So I'll just really quickly dive into MASH, which is a relatively recent addition to Maja's toolkit. It's a motion graphics toolkit, and it allows animation of multiple objects using mathematical equations rather than keyframes. So it saves you a lot of time. And I've created a new method of animating the lipid bilayer using this particular technique. And it's also great for creating large cityscapes, forests, gardens, and the like. So take a look at this. So what I've done here is created 3 small clumps of upper and lower lipids. I've animated them with MASH to give overall jittery movement. And then I've made the same 3 clumps animated with cloth to give individual motion to each lipid. And then I've constrained the cloth to the MASH. And so now you have sets of lipids with the internal and overall animation. And then I've used these 3 groups to make a massive dynamic MASH network, creating a large 1700 lipid bilayer. In hindsight, I think the movement's probably a little bit uniform, so I need to work on that a bit. So challenges and joys. In Australia, there are no specific biomedical animation courses, and there isn't really a set career path to follow either. I know that in the States and Canada, there are some amazing biomedical animation courses. And of course, you know, there are lots of design studios, biomedical animation studios offering, you know, a conventional employment path. So yeah, few job opportunities at present in Australia, but many internationally throughout Europe, the States and Canada. Biomedical animation can be painstakingly slow, and that can make it quite frustrating. My last animation, which was 14.5 minutes, took me a year to complete. So it is a very long process, as you've seen, because in the way that I outlined the process, I'm sure you would have some appreciation of how long it takes. I work alone. I'm a lone wolf at the Peter Mac, and that can be quite isolating. I can't turn to the person next to me and say, you know, what do you think of this colour scheme? Does this make sense to you? You know, can you see those molecules interacting? Constant problem solving. This is both a challenge and a joy. You have to bend the computer to your will. You have to make the computer do what it is that you want it to do. And you can go down many different pathways and reach many different dead ends trying to solve a problem and achieve an end. But when you do solve that problem, yeah, that is quite a joyful experience. So yeah, double-edged sword there. This is an absolute joy. I love combining my skills as an artist with my skills as a scientist. So being able to tell the stories of biology in a really beautiful and compelling way. It's very creatively rewarding because of that. And you have this beautiful end result that's very satisfying. So to summarize, biomedical animation is a powerful tool that makes the unseen cellular and molecular world visible. It enables the viewer to understand scientific concepts that might otherwise be really difficult to explain with text or words. And using visual technologies such as biomedical animation is really the best way to engage people with biology, given that we're becoming so incredibly visually focused. Visual technologies can be used to create a visual identity when presenting public health information, such as we've seen for COVID-19. And as we've also seen, a whole animation project follows a very particular process from start to finish. And that within that, there are a number of different artistic approaches that can be used to direct the viewer's attention and to facilitate understanding. I tend to use lots of dynamic simulations such as particles and hair and cloth, which are part of the sort of entertainment industry process. And I've started using MASH, which is a tool for controlling animation with procedures rather than keyframes. And although it can be difficult to get started in Australia, it's a very rewarding career with a beautiful, tangible outcome. And as I've mentioned, that's what gets me out of bed in the morning. I'd like to acknowledge Professor Ricky Johnston, Dr. Carol Gins and Professor Rick Pearson for seeing the need for biomedical animation at the Peter Mac. They've been instrumental in setting it up, which is fantastic. I'd like to thank my voiceover artist, Dr. Claire Fideli, my sound artist, Adam Hunt, my scientific advisors, and all of the people that have appeared in the live action footage throughout my animations. I'd also like to thank the Peter Mac Cancer Foundation who actually support me. And there is my Twitter handle and my email. If you would like to contact me, please do. And yes, the intersection of art and science is a wonderful place to be because indeed, art and science do meet in wonder. So thank you so much for your time and attention. I realise it's quite late at night for you now, so I really appreciate you taking the time out to come and listen to me, and happy to take any questions. Thank you.
Terrific. That was fantastic. So beautiful.
Oh, thank you.
The connection of art and science, and it's just wonderful. And one thing that struck me is that you know, you acknowledged all the people that help you, but you said you're a loner. So that's a dichotomy right there. So you really, you are, but you aren't, right? You have everybody there that you work with. So that's
You're
a
absolutely
wonderful thing.
right.
So,
yeah.
Terrific. Why don't you stop sharing your screen at the
Okay.
moment, and then we can, if somebody has a question, I will ask you to unmute. You could raise your hand or hit the reaction button. Who has a question? Sue Loomis has her first question. So Sue, you can unmute and ask your question.
Hi.
Hi there.
I was wondering if you had done anything with Cinema 4D.
No, I haven't used Cinema 4D, although I hear it's a wonderful program, and I
It
know a lot of
is.
biomedical animators use it.
Yeah.
I have
Yeah.
friends who use it. I think it's probably a bit easier, a bit more intuitive than Maya.
I think it is.
Yes, so I've heard. Yeah, but yeah, it's very popular in the biomedical animation world.
I've seen some of our researchers. I work at Massachusetts General Hospital, and I see some of our researchers using the strands and stuff, and I always wondered where they got them from. So I'm assuming they're taking them from that source and just, you know, using them without animation for structure.
Right.
So yeah.
Yeah, Cinema 4D is used a lot in the biomedical animation world, and I do have colleagues that use it. But yeah, I was trained using Maya, and you can get an educational version of Maya as well. And like you said, it is still the full version. But if you have a .edu email address, you can sign up for free access to Autodesk Maya.
Yeah.
So yeah. Thanks for your comment.
Great.
Okay, great. We do have a couple of comments and a question. I'll go to the question first. Catherine Hanlon is asking, what was the name of the app you mentioned in the beginning that does the hologram?
Oh, there actually isn't an app. It's like it's a technique that the contemporary artist Eric Zhong that I'm working with, he's developed it, and it's based on something that he's done in After Effects, but it's not an app as such. It's a technology that he's developed. So yeah, not sure whether he's up for sharing. I don't know, but yeah, it's not technically an app. It's something that we've developed. Well, he has actually.
You have to call him and collaborate.
Yeah, exactly. Yeah.
Any other questions?
If you want his details, I'm happy to pass them on. So just let me know.
Okay, great. Jamie Hayden.
Hey, Adam. Wonderful talk, Maya. Thank you very much for all that.
Thank you.
I actually had a very basic question out of this, because you're talking about the fact that there's not a lot of pathways for career advancement necessarily in Australia. How did you get there? You know, what was your pathway to get where you are?
Yeah, well, it was kind of a long and winding road. I started out as a scientist, went right through to PhD level, and then realised that a career in research probably wasn't for me. So I went into sales for a really long time, and then I met Dr. Drew Berry in the course of my sales work, and he's probably considered one of the world's leading exponents of biomedical animation. And it was just this, chance, serendipitous meeting. I've always been artistic, and I was feeling kind of quite, sort of bored and lacking creativity in my life at the time. And I was really trying to think of a way of bringing art and science together and struggling with how to do that. And so it was just one of these serendipitous meetings, and he showed me his work, and I was blown away and thought, yes, that's exactly what I want to do. So I set about retraining, did some formal courses, and then I was just kind of freelancing for a while. But the big break for me was getting a fellowship with Drew. The place where he works decided that he would really need to kind of take on a new person and train them up. It was actually part of a project called Visby Plus, and he collaborated with another woman, and the idea was to take 3 scientists and train them to become animators. And so there was myself, and there were 2 people in Sydney, and we spent about 2 years working on this, doing a couple of animations, and that was my big break. So that was where I kind of really consolidated my knowledge and got my level of expertise to that, that next level. And yeah, then I was able to kind of, I had to actually market myself to people. I had to kind of contact all these research institutions and say, this is who I am, this is what I do, and this is why you need me. So I had to kind of create it myself, which is quite difficult, because you really need to, find the champion who sees the need and the value in what you do, and you need them to kind of go into bat for you. So yeah, you can come up against a lot of walls, but when you find that right person who sees the value of biomedical animation, then they will make it happen, and you can make it happen together. So yeah, I actually approached the Peter MacCallum Cancer Centre, and we created a job, and I wrote my own job description and then had to interview for it, which is a very weird experience. And yeah, here I am. So yeah, you do, in Australia, you do have to really put yourself out there and try and market yourself and find the people that will listen. Yeah.
I was curious about the funding approach that you have. So you have a full-time job that's supported by the organization.
I do.
Versus trying to get independent funding for specific projects or working as a core facility or something along those lines.
Yeah, I'm very lucky in that because we're a cancer hospital, we have a lot of people that make donations and a lot of bequests and so on. As I'm sure you're aware, cancer is a very, most people are affected by it in some way. You either have it yourself, or someone that has it. So it's very, it's at the forefront in people's minds. So we have a very active philanthropy department, and so I'm supported by them, the funds that come through there. So I'm very lucky, actually. It's fantastic. But, you know, donors really like the idea of, you know, this defined enterprise that has, you know, something that they can see and that they can understand. So it's very attractive to them, which is great.
Anybody else? What my other question was, what about have you been approached, or has the institution been approached by, you know, occasional productions for children, for you know, to produce materials for education, for starting from kindergarten all the way through college? Is that something that you've been approached to, or they have been approached?
No, not me personally. I tend to make productions that are kind of pitched at years 10 to 12. So people with a bit of science. I'm kind of venturing more into the very basic stuff at the moment. But there is a person who works in radiology, and he makes animations for children, but they're not scientific. They're more about making children comfortable in the space when they come in for treatment.
Right.
So it's that kind of thing. So animations on walls that they can kind of interact with, and they feel welcome and safe. So yeah, we do have quite a lot of that sort of thing. Yeah, but not science for children as such. No.
Terrific.
Interesting question though.
Let's let Peter ask a question, and then we're going
Okay.
to go back to Sue.
That was a really interesting talk.
Oh, great.
So I'm a PhD student at Baylor College of Medicine, and I really want to kind of animate my work. I'm trying to understand like the function of this protein, and I was just wondering to what extent do you look at the research that is done in order to correctly portray the function or the movements? Because, like, you know, like when I think of the cell or the space around the protein, it's like, is there really that much space? And like when I look at models and papers, I don't, like, sometimes I see, like, I wonder, like, oh, is there a better way to portray that? I don't want to give misleading, or I don't want to convey the wrong idea on what this protein does or something. Like, I'm just wondering, like, to what extent do I have to like know the function in order to get these kinds of animations, or is there some imagination that goes into this? Or
Yeah. Well, as I discussed, I try to make my animations as scientifically accurate as possible. So all the molecules are scientifically accurate. What you see is actually how they look. I don't use simulation data to animate them. I know that there are people out there who do. What I try to do is I try to approximate Brownian motion. But no, I don't actually use simulation data as such. But yeah, you're right. There is a lot of stuff that you have to leave out. You know, when you're looking at the cellular environment, there are, like, you know, millions of different molecules that are all interacting. And you need to actually pair those away and pair those back and focus on the molecule that you're interested in. Otherwise, it's completely confusing and the viewer doesn't know where to look and they don't know what they're meant to be focusing on. So yes, you do have to tread a fine line between scientific accuracy and clarity. You have to have enough scientific detail, but not so much that you're bamboozling people. So yeah, there is a trade-off between those two things, and you have to decide what you put in and what you take out. But yeah, I mean, if you try to kind of make the cellular environment as authentic as possible, you'd have so much going on that the overall message would be completely lost. So that's how I approach it. Does that answer your question?
yeah, no, it's just, yeah, of course, nature is very complex, right? So
Yeah.
and very messy. So
Yeah.
There you go.
So yeah, you can just focus on the proteins that you're interested in, and I'd like to show some of the surrounding kind of molecular soup, and to kind of give everything context. But yeah, you really have to kind of focus on what it is you're trying to say. Yeah.
Okay. All right, perfect.
Very
Thank
good.
you.
Yeah.
So another question came to mind. You talked about the physical, what it really looks like, but do you ever have a need to animate like a chemical reaction going on, like a neural, like the neural pathways and things like that?
Right. Look,
How would you do that?
this comes up a lot and people do it all the time. I haven't had the call to do it as yet, but yeah, I'm sure I will. I'm sure that will happen. Yeah.
Chemotherapy and how it destroys the cells and what it does to the surrounding tissue and what it's actually interacting, how it's interacting, and visual. How do you show that visually? Right.
Yeah. We are thinking about doing a treatment animation somewhere down the line. I've got to get epigenetics out of the way first, so that will be my huge next project. So yeah, that is something that I would probably have to do. In that regard, I don't know, I would probably do it in a more schematic kind of way. I don't know that I would have that kind of going on in a cellular environment. I might kind of show it more along the lines of the sort of chemical equation kind of setup that that you're familiar with in chemistry class. But yeah, it's not something I've had to think about as yet, so I will now.
Okay, there you go. Great. Do we have any other questions? Well, how about everybody go off mute and let's give Dr. Maja a big round of applause. Thank you. Terrific.
Thank you very much.
Thanks so much. And
yeah, I really appreciate the opportunity to talk to you today. It's wonderful to talk to the Biocommunications Association. As I was saying to Peter, it's so lovely to be part of a family of people that have such a diverse and such a deep knowledge of all these different areas of visually communicating biology. It's wonderful. So thank you for the fantastic opportunity.